So again, thank you for coming, taking time out of your Saturday afternoon to hear about dementia. So I love this picture. Neuroscientists get really excited about this picture, and I'll tell you why. There are lots of different colors in it. It's called a brain bow. And that's because this is an area in the brain called the hippocampus. The hippocampus is absolutely critical for memory, and that's pertinent to our talk. And this is just a special kind of staining technique where we get to color every different cell, and someone thought that it would be cute to call it a brain bow instead of a rainbow. So my disclosures are that I'm the chief resident in the adult neurology program at Mayo Clinic just down the road here in Jacksonville, and I'm not reporting my opinions. I'm just going to show you the facts. So our objectives today, one, I want to provide an overview of the brain structure. You got to understand a little bit about what the brain looks like and how it functions before we can start to talk about what happens when things go wrong. And explain how the structure allows information to be relayed throughout the brain. So structure, function, and then dysfunction. That's where we're going to go today. All right. After that, we're going to talk about dementia. What is it? And what are, rather than the forms, I probably shouldn't have said forms, but what are some of the causes of dementia? And we're going to put an emphasis on neurodegenerative diseases, such as Alzheimer's disease, as one of the causes of dementia. And then we'll talk about what are the risks for dementia. And only after we've talked about the risk can we begin to talk about how you might be able to decrease that risk. Okay? So we got to talk about the risks before we can talk about prevention. And I promise I'm not looking at my watch for the time. I'm advancing the slides. So first, brain divisions. All right, I'm not going to go super deep into the brain, but I need you to know a little bit about it. A lot of times the brain is talked about like it's an electrical circuit. And that's true to a certain extent, and I think for our purposes we can kind of think about it that way. But we divide the brain up into divisions. In this case, this picture shows different lobes. So the frontal lobe is kind of the mission control center. It helps us in terms of planning and inhibition. We have the motor cortex, which is just what it sounds like. It helps us to move. We've got a sensory cortex. We've got the temporal lobe that's important for hearing and emotions. The occipital lobe is very important for vision. The cerebellum is an area in the back of the brain that's very important for coordination and keeps us from wobbling when we walk. And then the brain stem is an area that's very deep into the brain, and that's where a lot of the primitive functions are. That's the part of the brain that helps us with respiration, with regulation of blood pressure, and a lot of other things that fortunately we don't have to think about controlling because the brain just handles it on autopilot. Now, we divide the brain up even further. As you'll see up here, all these different numbers refer to different areas. And on the surface of the brain, we understand a lot about how things are divided up. In fact, the motor cortex, which is supposed to be represented by this purple strip, and then the sensory cortex divided by this blue strip, we actually know that areas correspond to moving the hand. Areas correspond to moving your tongue. There's actually, ironically, there's a big part of the brain that's important for moving the tongue. So that's just a little sneak peek of how the overall structure of the brain looks, but we can go a lot deeper than that. And it's really proper integration of all of these areas that's absolutely critical for normal function. So this is a slice of the brain. So I want you to think about the brain as kind of a salami. And when we use MRI, we take slices through the salami and we get to look at each one of those slices and understand more about the brain structure. Okay? So this is if you're looking straight on at someone and we're slicing the salami backwards. Okay? So this is the left side of the brain. This is the right side of the brain. This is the top. Now remember that brain bow on the very first slide that I showed with the hippocampus? Here's your hippocampus. Right in here. You have two of them, fortunately. Okay? So let's zoom in a little bit more and see if that shape starts to look familiar. Well, not really. Not yet anyway. Let's zoom in a little bit more. Does that shape look kind of familiar there? Okay. Now, just so you know, if I were to cut into your brain, there's not neon purple and neon green and neon blue. That's fluorescent dye that we use to be able to look at neurons. But each little dot that looks like an ink splotch has some lines coming out from it. Those are individual brain cells, the neurons in the brain. These are the guys that do all the work in the brain. Okay? So we have technology to look even further at these neurons. And this is what they look like. They're gorgeous. So this is our cell body, and it's got all these little projections that come out. Okay? They're like wires, like cables. And those cables can contact with other cables. And we have extensive connections here. You'll notice that along these cables there are lots of little dots. Can y'all see those dots there? The bright yellow dots? Each one of those dots is a connection point from another cable, from another neuron. So we're just looking at 1, 2, maybe 3, 4 neurons down here. But just in this small little area, you can get an idea of how integrated and how complex the system is. But we're not done yet. We can go further. Let's look at one of the cables. This shows that, see kind of the studding along the cable? Each one of these little dots are those contact sites from another neuron. So just on a very small stretch of the axon, we would call it, or the dendrite rather, we would call it, is getting a lot of inputs from other neurons. We can go even further. Let's look at what those inputs look like. This is using an electron micrograph, or an electron microscope, that works a lot like sonar. So sonar that might be on a ship or a submarine, where you bounce sound waves off something, and based on how fast those sound waves come back, you get an idea of how far the other submarine is. Okay? With this technique, we use electrons, so tiny electrons, and bounce them off of structures, and based on how quickly they come back, we can develop an image. So what you're seeing here is one of those little contacts, and here's another one. And they speak to each other through this little gap. Alright? Now you'll notice that there are lots of little balls in this. Can y'all see the balls there? Each one of those little balls is filled with chemicals, neurotransmitters, things like glutamate, serotonin, dopamine, GABA. And it's at this gap, called the synapse, that those chemicals are released. And so they come out of the synapse and then they float across, almost like a baseball coming out of a pitcher's hand into the catcher's mitt. There are very specific baseball gloves, so to speak, on the receiving neuron that catches those neurotransmitters. Now, what causes those neurotransmitters to pop out? They just don't pop out at normal, but you have electrical current that travels down all these wires and causes the neurotransmitters to come out, dump out, and then be picked up by the baseball glove, so to speak, on the other neuron. And then that causes more current, electrical current, in the receiving neuron. So we've got electricity, chemicals, and that's how everything communicates in the brain.